This is a production of Cornell University.
Thanks. Yeah, so I'm with Breeding Insight. We're under biotech. We're just up Tower Road next to the Boyce Thompson Institute in a small building that you probably haven't noticed before. But we're on campus if you're looking for us. So yeah, super excited to share the work that the team does at Breeding Insight. Borrowed a lot of slides from people, and we'll get to talk through these activities. So overview, definitely spend a little bit of time explaining what Breeding Insight is, why we were founded, what we're working on, go into some specific examples of genomics projects and how we're assisting breeders, talk about some bioinformatics results, talk a little bit about some of the image analysis projects that we've worked on with breeders, talk a little bit about Delta Breed, which is our database management platform, and then I've got a couple miscellaneous topics at the end. So Breeding Insight, the mission that we were given was to help transform breeding by enabling the implementation of genomic insight and selection as part of routine specialty crop and livestock breeding across all of USDA ARS. So I think what people at USDA ARS were seeing and what we've discovered working with a lot of specialty species is that a lot of the challenges in corn and soybean, a lot of the tools and items that have been deployed aren't readily transferable to specialty species in a one-to-one capacity. And I think this list kind of shows that with corn and soybean, you've got very clear full mechanization. We're looking just at 2 annual species. We've got functional diploids. We just have to deal with seed components, seed planting. A lot of commercial breeders are in this space, primarily focused on high yields, and you get to have dry harvest. If you try to transfer that list over to blueberry and lettuce, it falls apart pretty quick. So again, how do we create tools? How do we adapt these tools and service breeders when this is not the conventional list of things that have been made for corn and soybean? All right, so the breeders that we work with, breeders are tasked and asked to do a whole lot of things. So to have a successful program, to make good progress, right, you've got to have good breeding program management, a lot of good field data collection tools, store your traits properly, manage all your samples, whether that's DNA samples, tissue samples, seed samples. Again, can you work within the USDA germplasm banks to bring in new and novel genetic diversity, identify new traits that are plaguing your growers, storing your genetic data, perform genetic analyses, and then make all those decisions and report out. So breeding's not a sole enterprise. I don't think any single person can do all of this. So what we're really looking at doing is how can you create a good team? So I think any breeder today would say, I would love to have a great bioinformaticist. I'd love to have somebody who's knowledgeable of genomics. I'd love to have a good statistician. And maybe I'd like to have a software developer to help connect and build new tools to serve my program. So to provide this for every species, for every breeder across USDA ARS would be extremely challenging and extremely cost prohibitive. All right, so where Breeding Insight kind of fits within this scheme, again, there's a lot of basic research that comes out, super new techniques, new research coming out all the time. Plant and animal breeders, as I just showed on the list before, they're tasked with a whole bunch of other things. They don't always have time to go figure out the newest software, figure out the newest genomic thing. Is it going to disrupt their current workflow? So that's where we kind of fit in, is kind of looking over at the basic research, hearing directly from plant and animal breeders, what are their needs, what are their challenges, and help ease that technology adoption into their program. So if they say, I want to start using genetic markers in my program, that's great. Let's talk about what you want to use them for. How many do you want to deploy? How are you going to use it? All right, now let's show you a suite of genomic tools that might be a good fit for you. Let's talk price. Let's just kind of narrow down the search space for you so you're not wasting your time chasing a bunch of different things that disrupt your current workflow. Let's give you a really high probability of success for what you want. All right, so a little bit more about who we are. So again, we are a USDA ARS funded initiative. We're located at Cornell University. We are Cornell employees, so that's really nice at the moment. We connect and build resources for specialty crop and livestock breeders. So we'll talk examples for both. And as I just mentioned, we really picture ourselves as a service and technology transfer model. Again, just helping provide additional support to USDA specialty breeding programs. And I know I'll focus a lot on science team developments, but want to highlight that we're a team made up of scientists as well as software developers. All right, so a little bit about what our growth has been like since 2018. So the idea of starting Breeding Insight and the inception came in 2018. We got our first group of species in 2019. So there's the 6 species: trout, salmon, alfalfa grape, blueberry, and sweet potato. That represented about 15 breeding programs across the USDA ARS. In 2022, we got 10 new species, pictured down at the bottom, and also the formation of Breeding Insight. So this is a group doing similar work to what we're doing, but they're primarily focused down in the Southeast. And they have a slightly different research model than we do. In 2024, we got 11 new species pictured on the right, and that brings us up to supporting around 51 breeders and breeding programs across the US. All right. So here's kind of what it looks like, where all those breeding programs are located. So you can see we've got some high capacity over in the Pacific Northwest, in Corvallis, Oregon, as well as Prosser, Washington. Also quite a bit of capacity in Madison, Wisconsin, a number of species there, and then a decent number in Beltsville, Maryland. So really the idea is build some capacity at Cornell University, and then we can serve as kind of surge support for bioinformatics, genomics, phenotyping, software development to these breeding programs across the US. One of the things that's really fun with working with all these different species is we'll hear of a solution or a challenge in something like trout, and then we'll hear a similar thing in cranberry and take a tool from trout and be able to apply it in cranberry, where those two groups would never meet at a typical conference or discuss these ideas. So we sit at kind of an interesting spot, hearing a lot of just breeding technology that's coming through, and we help funnel and distribute that information out to other groups. All right, so that was a little bit about Breeding Insight. Let's talk about some of the other things that we're working on. So one of the big challenges that we were set out to do with looking at our species portfolio is we can't constantly be building one-off solutions for every species or every program that we work with. So talking with breeders, working with breeders, a lot of the challenges are the same, but trying to figure out what is a common solution that's going to work across all those species is a challenge, but that's what we're working on, and I think we're making good progress identifying a number of tools and services that work in that capacity. So again, what we refer to this as kind of creating species agnostic tools. Can we identify something that can help breeders perform their own bioinformatics regardless of the ploidy level of the species, regardless of the marker platform that they're using? If a species says they want to use genotyping, can we find one good genotyping provider that can fit the needs for all the species that we're working with? Same with phenotyping. Can we find something that works on berries and also works on leaves and also works on roots? Kind of simpler solutions. And then the database management, that's the same component. Can we find a way for breeders to store data for annuals as well as perennials for things that are at the plot level, things are at the plant level? So just really try to create species agnostic tools across all of these species. All right, we'll go into some specific genotyping services, tools, and projects. So here's what I talked about, the challenges with genetics, looking at all the species that we work with. So we're going all the way from a haploid diploid system in honeybee through a lot of diploids, with some of them having sex chromosomes. And then we've also got some in fish breeding. They will change the sexes of the fish periodically. So that's a fun challenge. And then if you can look across our polyploid species, we've got tetraploids all the way up to 12X species. And not always 12X, sometimes other things. So I think as we keep looking at this stuff, it keeps getting more complicated, and some people say more fun. All right, so some additional challenges with polyploids is that you usually don't have a high quality reference genome due to the complexity of the genome. It's hard to get accurate dosage calling, and it's hard to phase the markers, especially for polyploid species. So those are some of the challenges that we're facing. All right, so kind of that species agnostic tool that I talked about. We've identified DART, Diversity Array Technology, as a company in Australia that's working really well to fit a lot of the needs and requests that we have. So this is our current model. I'll talk about the marker technology from them on the next slide. But breeders that come to us that say they want to do genotyping, we'll work with them to create a species-specific marker panel. And then the breeders in this equation are they're doing the tissue collection on site. So we work with them to provide best practices, what's the plasticware they need to do, how do they ship it, how much to collect, what are they working with. We help them with the shipping to Australia, which again, they've got strict borders for bringing in outside material. The DNA extraction is also done at DART, so that's about $2 a sample, which is great. And the actual genotyping, depending on the species, $9 to $11. So pretty low cost, pretty high throughput. DART's been great to work with, and that genotyping data can come directly to us. So we can help the breeders QC the data and perform any early bioinformatics analysis that they need, and then we can push that back to them, and then they can make direct breeding decisions with the data. All right, so just a little bit about the DART tag is the technology that we're using. That's the, so it's a targeted genotyping by sequencing platform based on this padlock technology featured on the right. So we can target any SNP or small indel into the DART tag panel. Typically, we're making the marker panels to target 3,000 loci. That's within a single primer pool. And what's nice about the technology is that we're allowed to force in any major QTL or markers used in marker-assisted selection that the breeders are already incorporated into their breeding program. So we can force those in, and then we try to round out the rest of the panel with genome-wide distribution of the markers. All right, so we're hitting the same 3,000 loci every time, so we get relatively low missing data, and we found that this works plenty well for any genetic maps that we want to create, any QTL analysis, any GWAS, and we're getting good genomic prediction accuracies for most of our species, for most traits. All right, so yeah, we've been busy making a lot of marker panels. So here's the list of species that we currently have. So we work with DART to make these marker panels open source and publicly available. So if you're an alfalfa breeder, you can contact DART and use the same alfalfa marker panel, whether you're in industry or whether you're USDA ARS. So that data has a one-to-one comparison. Yeah, so here's a number of species that we've worked with. Here's the ones that are currently in development. So yeah, we'll have a new hop marker panel coming out. Watermelon, red clover, hemp's being worked on with the group up in Geneva. And we've got blackberry being worked on out in the Pacific Northwest. So again, just to reiterate, what we've seen with the DART tag marker technology is that we found it to be readily accessible. We get good communication with DART. The breeders are also able to easily send their samples. The data coming back is highly reliable. We're getting fast turnaround time. The price point's quite affordable, high repeatability among the calls that are being made, and we like that it's a targeted marker platform, that you're hitting the same 3,000 loci. Right, so a little bit about the data that you get when it comes back. So I mentioned that it's you get targeted sequencing. So we're targeting a SNP of interest. So here we're looking at this G to A, but because it's targeted sequencing right now at 81 base pairs, we'll also pick up these off-target SNPs. That's what we're calling them. So for any loci, we're getting additional, what we're calling microhaplotypes. So instead of just biallelic SNP calls at the locus, we're getting much more information. So more similar to SSR markers. Okay. All right, so some additional information on why we like DART tag for genotyping for the species that we work with. Again, same set of loci across all projects and samples. So it just makes sharing information, particularly when there's multiple breeding programs for a species. It works really well on low quality DNA. We get low missing data. We get pretty good read depth at around 50 reads per locus per sample. So this really helps with multi-state dosage calling, especially for autopolyploid species. We like the multi-allelic reads, what we call microhaplotypes across the target loci. And then, yeah, linkage maps constructed using DART SNPs can identify some genome assembly mistakes. So we've seen this in cranberry and lettuce. All right, so getting multiple microhaplotypes per locus means we've got a whole lot of loci. and a whole lot of genetic data that we need to keep track of. So here's some examples in alfalfa on the upper left. So again, if we were just looking at biallelic SNPs at 3,000 loci, we would expect to see 6,000 alleles. Okay, we're identifying 32,000 unique microhaplotypes, so coming in at around 10 microhaplotypes per locus. There's just a lot of additional information at those loci that we're targeting. So we want to keep track of that. We want to work with our breeders, keep genotyping a number of samples, see if we can get kind of where do these stop? How much can we capture enough diversity with this single marker panel that these kind of asymptote? And we can say, yeah, we feel like we've identified 90%, 95% of the genetic diversity for the loci that we're looking at. So across the 7 species that we've currently got marker panels out for, we've got 156,000 microhaplotypes. Yeah, so we'll talk next about setting up databases to keep track of this information. Right, so again, so here's the target SNPs, the off-target SNPs. So again, we're working to establish the best workflows for filtering and processing this genotype data as it comes in. We want to create file formats that work with other applications for downstream analyses. So we want breeders to have flexibility if they want to use tools that we're not developing in-house. Can we get them the data file formats so that they can go run their regular analysis that they've already set up? Yeah, and then we're trying to create and test internal apps for user-friendly pipelines. So we're working on something called Haplotype DB. All right, so here's a picture of a component of that Haplotype DB. So this is called HaploSearch. So this allows you to query those microhaplotypes. So if you want to look at a particular locus to see how many microhaplotypes you have, or if you've identified a microhaplotype associated with a major QTL, you can go in, look further into that allele, the microhaplotype, and look at other germplasm that might have that same allele or QTL. Or you can also look across projects and say, I'm only seeing a little bit of diversity at this one loci that I have. I'm seeing a lot of other projects have a whole lot of diversity. Maybe I should reach out to my collaborator and ask for some germplasm so that I can increase diversity in this targeted region. Okay, we'll shift to bioinformatics. Right, so this was a fun one, alfalfa creeping rootedness, QTL. So the breeder that we were working with had identified this as kind of classic alfalfa, kind of staying within its planted row very nicely. He was very interested in what's going on with this spreading alfalfa that he had identified, thinking that maybe this would lead to increased tolerance for biotic or abiotic stresses, or maybe there could be a niche market for growers who want to plant this and just let the alfalfa spread in the field. So we genotyped an alfalfa population, segregating for the trait, using the 3K DART-tag marker panel for alfalfa, performed GWAS poly within this big app, and I've got a slide, I'll talk a little bit more about that. But yeah, we identified a major QTL on chromosome 6, explaining this trait. So we can share that back to the breeder and see what he wants to do with it. All right, next project, genomic prediction. We're going to stick with alfalfa here for another minute. So alfalfa, when we started working with them a few years ago, they didn't have, they really weren't using a lot of genetic tools in their analyses or in their breeding programs. And now that they have a marker panel, they're very excited and they're talking about moving into genomic selection. So they're really interested. They're kind of in the testing phase right now. Like, what accuracies are we getting for our traits? How do we create a training panel to do this? And so we're working with them to show kind of like what's the diversity with what they're working with, what they're starting with their training group. What percentages do you need from all these different subgroups to get the best predictions that you have? So I think, again, a lot of other species, plenty of literature on this. Alfalfa is just coming to the table now. So trying to assist them with this. And we can see that we're getting pretty good prediction accuracies for things like height in their program. All right, the next one. So it talked a lot about microhaplotypes, having these multiple information per loci. So thinking about, can we squeeze more information using that data than just biallelic SNPs? So what kind of genomic prediction accuracies can we get if we actually use the microhaplotypes for pedigree relatedness versus just the biallelic target SNPs? So we're seeing slight increases in there, and we're really interested in exploring that more. All right, so just alluded to this big app. So that stands for the Breeding Insight Genomics app. So this is a collaboration with the genomics and bioinformatics team, as well as other key people. So trying to make our Shiny app, just to make a web-based application to perform genomics and bioinformatics analysis that's easily accessible. Somebody can use it with no coding experience. It's got really good integrated help. We're working on putting out tutorials. There's help pages online. There's also clickable information within the app to find more information, and hopefully you can feel confident running your own analyses. And then also if you use the tool, you've got the citations for what tools and additional applications were incorporated. And again, broad support. We've got the ability to pick what ploidy your species is within the app. So diploids, polyploids should be able to use the tool without any issues. Okay, yeah, so the basic analyses in there are, again, we can do dosage calling from the files coming from DART. You can look at your data using PCAs, do some basic genetic diversity analyses. You can perform GWAS, and you can do genomic prediction. All right, let's talk image phenotyping tools that we've had. So this was a sweet potato nematode project. So this one was with the sweet potato group down in Charleston, South Carolina. So they were doing root washes of potatoes to look for which ones are most resistant or most susceptible to nematodes. The classic model was to dilute these root washes, sit at the microscope for 3+ weeks, hand counting the nematodes on the screen. That seemed fairly inefficient. They reached out to us, could we come up with a better solution? And so tears in the audience, and this was our first application of doing machine learning for image analysis. So it worked really well. So you can see on this slide, there's still a lot of plant debris, but it's picking up the nematode eggs very clearly from that plant debris in the background. And looking down here at the F1 scores, so the models agree more with humans per counts than one human to another human. So pretty nice. The scalability of this, again, going from single slides to now you can pipette 96 samples into a plate and then run it through the machine and get your images really quickly. All right, the next one, this was the trout image one. I'm sure we've got Edwin online. We've also got AJ in the audience. So this came from the trout breeder in Hagerman, Idaho. So he was really interested in taking images of the trout eggs following fertilization to identify which ones had successful fertilization versus which ones were blank or aborted. You can see the differences with your eye. The initial challenge was that the eggs are quite glossy and shiny. So we had to come up with a imaging system to reduce that glare on those to get clear images so that we could actually categorize them. So the efficiency is that these trays with the eggs sometimes have up to 7,000 eggs on them. And this machine learning model will analyze it within seconds with minimal handling. Recording over 95% accuracy on dead versus viable. And the scalability is that they worked very nicely to create this very simple light box and deploy that in a system that needs to stay dry, can't have a lot of handling, and just needs to be readily deployed on site. I think this is the final one for image projects. This was potato growth defects with Boja. So this came out of the Max Feldman's potato breeding group in Prosser, Washington. So he asked, can we segment and measure negative growth traits on potato tubers? So his system is that he can harvest them, wash them, put them on a conveyor belt, and then they get automatically imaged, and then the potatoes are cropped out. So he wanted to identify some negative growth traits, like this growth crack on the tuber, and then also these sprouts that are on the tuber. So again, the efficiency is hundreds to thousands of tubers can be individually evaluated in an automated run, and we're getting 90 to 94% accuracy, where this is now replacing the classic 1 to 5 rating for negative growth defects. All right, talk a little bit about the data management software called DeltaBreed. So here's our logo, here's the name. Yeah, so we're definitely trying to abide by fair data practices. So make the data easily findable, accessible, interoperable, and reusable. So we try to adhere to the community standards set by BRAPI, so that's the Breeding Application Programming Interface. We talk extensively with Pete Selby on that type of work and try to stay up to date and help provide information back to the community when we can. Again, the big challenge is creating things that are general, but also can be specific to breeding programs at the same time, as we're trying to manage a lot of different species. So we've recently released the Delta Breed version 1.0. We're considering this our MVP, or minimal viable product. So with that, we know that we're working with pretty basic features, kind of data in, data out. There's known limitations, and at the moment, we're trying to have a controlled rollout to our breeders. So we're really just trying to support the USDA ARS breeders that are most engaged with us and have the most immediate need for data management support. Along with that, again, just try to really help get documentation and training resources in place so that they can do additional learning and know best practices. Right, so with that, some things that the system can do at the moment is we can set privacy and administration levels for each user within the breeding program. You can upload germplasm, you can upload ontology, so that's the traits that you're going to phenotype with. You can upload experiments and observation data, and then we've got the BRAPI connection to push data to Fieldbook. Right, so yeah, users sign in using their ORCID, so that your user permission levels associated with that information. So here's an example of germplasm records. So everything that comes in gets a unique GID, the name of the germplasm, and then you can track the pedigree information, and you can always open and expand to see more details on that individual. Right, pedigree viewer. So this was a BRAP coming out of the BreedBase group. So this was an example of Zuni. So this was a pecan release down here, and they wanted to track back and see what were the parents and how did they get to it. Right, so ontology, this is, again, phenotypes, ontology. What's the set vocabulary that you're using to go and collect phenotypes out in the field or in the lab? So again, a shortened name here, you can include the full name. What's the trait that you're measuring? What's the scale class? What's the units that you're working with? If you click show more details, you get a little bit more information for what are each of the values that you've set 1 to 5. So again, the idea is that you're providing enough information that if I gave this to somebody to go out to the field to score, that you would feel confident that they could go score correctly, that they're not going to invert the scales or invent something new. So again, something as simple as plant height is, well, am I measuring it in the middle of the season? Am I measuring it in the end of the season? Am I measuring it at the top of the flower, to the flag leaf? So just being very clear on what's the defined vocabulary, and that's where we come in with the trait method and scale. Right, experiments and observations, I think pretty classic, your germplasm name. There's the GID that's associated with that one. Is this a test plot or a check plot? What environment are you working in? What are your row and column positions for that? You can set lat and long if you're using a GPS to track where those are in the field. A unique observation unit ID. This is system-wide for that data row. And then fill in and upload your phenotype data for the experiment. And then downloading data, again, you can decide, am I pulling all environments associated with that experiment, and what's the file format that I would like my data to come out in. All right, so the connection to Fieldbook via BRAPI, this has worked nicely. We've got a QR code on the screen for your breeding program, which will push the data into Fieldbook so that you can indicate which field you want to go collect data for and upload your phenotype traits as well and select those. And then you can go out in the field and digitally collect your phenotype data. So hopefully minimal downloading of files and re-uploading the files. You can just scan the QR code and push that using BRAPI. All right, so an important component not to be missed in creating all of these tools is also the training that needs to go into it. So we have a learning hub online so that people can go and find this information. We can create links, provide SOPs, plenty of Zoom instructions, plenty of meetings with breeders to hear what are the challenges, how can we better help. And when we can, we love to go out and have in-person site visits with our breeders to really hear firsthand what are they faced with and establish a good connection with our collaborators. Right, additional software demo applications. So we've got YouTube videos coming in pretty consistently now to help talk about the Delta Breed software, the Big App, and other tools that we're developing. Okay, I think we've made it almost to the end. Just a couple fun miscellaneous things to finish up. So we have a long list of species that we work with at Breeding Insight, but also just taking a look at what CALS already works with and Agritech up in Geneva. There's about 50% overlap between these groups and the species that we support. But we also have our own set of species that aren't common between the two. So again, I think we hope that there's tools and services at Breeding Insight and that these can be transferred over to the Cornell community. All right, just also want to take a moment to highlight that Breeding Insight and BI OnRamp received the 2024 USDA Secretary Honor Award. So this was, I think, category 4 for providing all-American safe and nutritious food. So this was on January 10. So Dong Yen got to go down. Tim Reinhart is our PI representing the Office of National Programs at the USDA, and Keo is with BI OnRamp. So they got to go down and enjoy the celebration with Tim as we won this award. All right. Final slide. Just want to highlight a number of Cornell trained individuals on our team who were trained at Cornell. So thank you to people who helped train these great people. Glad to have them back in the Cornell community, providing more tools and services and sticking their whole careers with plant breeding and genetics or physiology. So thank you so much. But I think that's a pretty good number. All right, and definitely a lot of people to thank. You know, we have a ton of support with the BI OnRamp staff, the ONP, a lot of people on the Breeding Insight team at the moment, and thanks to our advisory board for helping answer questions when we have them. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.